Quorum sensing is a mechanism that prokaryotes use to communicate with each other based on cell density. Quorum sensing often initiates group behaviors in cell colonies. Prokaryotic organisms sense population density by emitting chemicals called autoinducers that can be recognized either between cells of the same species or between different species. Microbial quorum sensing is similar to how surprise parties work. Before the party begins, all participants are waiting for the right moment to spring the surprise. After a certain density of people has been reached, the party can begin. Cells constantly emit as well as receive signals to communicate with other cells. Consequently, the extracellular environment contains certain levels of cell-specific autoinducers. The number of signal molecules known as autoinducers is proportional to the amount of cells in the environment. Once a cell's receptors reach a certain level of signaling molecule saturation, biochemical pathways may be initiated. These pathways often involve gene activation or deactivation. Thus, just as the actions involved in the surprise party only begin once a certain population density has been reached, cells also wait until after a certain density has been reached before initiating group behavior. Autoinducers trigger a pathway of biochemical reactions that modify gene expression, initiating group behaviors such as bioluminescence, biofilm production, virulence, and sporulation, among others. Many cell behaviors such as bioluminescence and biofilm production are only useful if many cells are involved, since single cells can neither produce significant quantities of light to be noticed in the environment, nor sufficient biofilm to allow group adherence to a surface. There are two different types of autoinducer molecule. Gram-negative bacteria use acylated homoserine lactones, whereas gram-positive bacteria use processed oligopeptides. Both signal molecules depend on membrane properties for dispersal from and entrance into the cell. Cell membranes composed of lipid bilayers are impermeable to proteins, but permeable to small nonpolar molecules. So in gram-negative bacteria, whose autoinducer is relatively small and nonpolar, the amount of autoinducer in the extracellular environment is the same as that in the intracellular environment. However, in gram-positive bacteria, autoinducer accumulates in the extracellular environment and is bound to receptor membrane proteins rather than passing straight through the gram-positive membrane. The bacterium Vibrio fischeri provides the best understood model of quorum sensing for gram-negative bacteria. The Hawaiian bobtail squid utilizes the bioluminescence of Vibrio fischeri in order to conceal itself or attract prey. Generally, gram-negative bacteria have a regulatory protein that produces autoinducers as well as another sensory regulatory protein to bind to them, triggering a gene regulation response. However, some exceptions utilize a two autoinducer sensor pair for quorum sensing regulation of more than one gene. The soil bacterium Bacillus subtilis provides another model of quorum sensing for gram-negative bacteria. Bacillus subtilis uses quorum sensing to trigger spore formation to survive through harsh conditions. Gram-positive bacteria encode their autoinducer in their chromosome. Transcription and translation of the autoinducer gene produces a peptide that must be modified by cleavage. The cleaved peptide can be secreted by the ATP binding cassette transporter to transport the oligopeptide out of the cell. When an oligopeptide binds to a sensor kinase, a phosphorylation cascade occurs, resulting in upregulation or downregulation of certain genes.